Well, welcome everybody, and Paul. That's a special pleasure to have him with our senior college group. I did some research to prepare this introduction, filling in for Joanne at Sick Kids, where Paul is based. You've all seen his bio on our SC Talk information, but I wanted to look more into his Google profile to see what he's been up to lately. He clearly has tens of thousands of citations, but remarkably, they're still growing after 20 some years. His citations have increased. He has colleagues all over the world. And he has now well over 10 review papers that are highly cited. So how did he get there? I mean, I saw him as a student when he arrived from England, when he was more interested in soccer than in science. You know, you make friends and you score goals. It's very easy. Well, science is now a team sport. We all share colleagues, we make friends. And then if we can share methods and skills, we can solve big problems. And that's what Paul has been so good at doing. When he arrived in my lab, he met people down the hall and started collaborating, even with my colleagues more than I did. You know, I'm a tennis player, so it's an individual sport, but science is now a team sport. He went to UCLA and met people from all over the world with new techniques to visualize brain neurons and synapses using genetic tools. And that was remarkable. Played soccer with a lot of them, actually, still does, when he manages to see them on his jet set travels. So what's remarkable is this was in the sub-basement of Sid Smith, sort of a dungeon. He not only met the colleagues, but he met his lifetime partner, Sina Jocelyn, who many of you have heard. And she became his greatest colleague and asset. So I decided to go to their lab last month to see how the pandemic has affected them. And to my great surprise, they seem to be more active than ever. Over 20 students, all working together each day as sort of equal colleagues. Every one of them wanted to show me their latest pictures of neurons lighting up, showing memories going on and off. Paul says that now that he's not a jet setter, he can spend more time with his students and his daughter. That's just remarkable to see how it's worked out at Sick Kids. We were so happy when they hired both of them from the United States together to come to the University of Toronto. And so many of you know how that's worked out for all of us. So his topic today is his latest review paper, which I hope you can read, on the persistence and transience of memory. This shows how human biological memories are related to artificial intelligence. And this big picture is something that I could never have imagined. The biggest question in neurosciences is how intelligence and memories are formed step by step, neuron by neuron, gene by gene. And that's what's in this paper, which you can get more of if you have that opportunity. So welcome, Paul. You are my persistent and memorable friend, and I hope you get to share all of your work with my many friends at senior college.
Thanks very much, John, for that great introduction. Of course, I will never forget the dungeon of Sid Smith and my sort of formative years I spent there. Okay, so I'm just going to share my screen. I think this one should work. Let's see. Okay, yeah, and okay, so as John mentioned, the title of my talk is The Persistence and Transience of Memory. And so the slide's not advancing. 1 second. I try just to share the screen again. Let's try this one. Okay, now we're good. Okay, so yeah, the persistence and transience of memory. So as a neuroscientist, or even not a non-neuroscientist, you might define memory as the transmission of information through time. And then you might imagine that an ideal memory system would be one that exhibits perfect persistence. That is a memory that, that transmits the greatest amount of information with the highest fidelity across the longest stretches of time. But the few examples we have of individuals with such ability, or something which resembles that kind of ability, suggests that remembering comes at a cost. And so this is Solomon Shereshevsky. He grew up at the beginning of the last century in Moscow. And when he was about 20 years old, he joined a local newspaper as a junior reporter. And at that newspaper, each morning, the editor would gather the various reporters into the editorial room and would give out assignments. And in giving out the assignments, he would detail each of these assignments in terms of the events that they were to report on, the people, and the places in which the events happened. And during these meetings, all the reporters would take diligent notes, apart from Solomon Shereshevsky. But despite that, he, when his editors would ask him about any of the assignments, he was able to remember and catalog those assignments with exquisite detail. So the editor became sort of amazed and struck by this amazing memory ability that Shereshevsky had, and encouraged him to go and see a friend of his who was a psychologist named Alexander Luria. And then in Luria's office, this is the type of thing which would happen. So Shereshevsky was, or patient S as he became known, was given lists of numbers like these, and these sort of random numbers, and he was given a few minutes to study these numbers and then asked later to reproduce them. And he always reproduced them without making any errors whatsoever. So this memorization ability didn't seem to be limited to lists of random numbers. If you asked him to do lists of letters or lists of nonsense syllables, or randomly chosen words, he was equally good. And it didn't really matter how long the lists were. He always showed perfect recall. And it also didn't really matter how long there was between learning the list, the study sort of phase, if you like, and being tested on the list. So he showed perfect recall, whether tested minutes or hours or days or weeks, or even in some cases years later, he could show a pretty good recall on some of these sort of arbitrary lists that he'd been given. So Luria spent much of the next 30 years or so cataloging patient S's memory. And he summarized his findings or his observations in this book, which has become a sort of famous book in the memory field. It's called The Mind of a Mnemonist, a little book about a vast memory. And in this book, he concluded about patient S, he said his memory has no distinct limits and for all practical purposes was essentially inexhaustible. So this sounds great. He's got perfect memory, but he also noted that this seemed to come at a cost. So what was the cost? So when he gave him similar lists like the one just shown here, as before, you'd expect that he could memorize these lists perfectly. And of course he could. It made no errors when tested on this. But at the same time, he was completely oblivious, apparently, to a pretty obvious pattern within that number series. And that was the case whether it was this number series like the one shown here, or if he was given lists of words to remember, like these ones shown here. He could reproduce these words without missing any of them, making any mistakes whatsoever. But he was seemingly blind to the fact that the words were linked in meaning. So obviously the top set of words are all types of birds, the bottom set of words are all types of liquids. And so the cost seems to be, well, you've got this great ability to memorize, but the cost seems to be an inability to detect patterns, if you like, to abstract from these data. And so what that suggested to Luria, but also to others as well, is that this type of forgetting, at least partial or some kind of controlled forgetting, might be an important component of a healthy memory system. So despite that, over the last 100 years of neuroscience research, neuroscience have mostly been asking questions about how do we remember? That is, what are the neural mechanisms of persistence? What I'm going to talk about today instead is a much smaller literature, and probably from a neuroscientific point of view, a much more recent literature, and that's on the basis on how we forget, or the neural mechanisms of transience. And in the first part of my talk, what I want to focus on in particular is the role of hippocampal neurogenesis in forgetting. And so I'll talk in more detail about what hippocampal neurogenesis is later. And it's one of several mechanisms that have been identified that seem to contribute to forgetting. And then the second part of my talk, I'll talk about why we might forget. And that is, what is the adaptive value of controlled forgetting? So you can imagine the benefits of persistence, of remembering, are quite obvious. But why do we forget? Why is that useful? And it sort of goes back to patient S. It seems that it optimizes memory-guided decision-making in changing and noisy environments. So basically, a little bit of forgetting is good because it allows you to abstract, to detect patterns, and so on. So first, let's think about persistence. And we might define persistence, or talk about persistence in the following way. You might say remembering requires that some aspect of our current brain state reflects a past brain state corresponding to a specific event. So that's like a very sort of formal way of saying it, but maybe a simpler version of that would be that remembering involves reactivation of the patterns of neural activity that were present during encoding, during the initial experience. And so when we experience things, that neurons are active. If they are reactivated at some later time point, perhaps that corresponds to the psychological process of retrieving that memory. So here's a textbook version of how that might work. And so this is a very sort of abstracted, very simplified neural network. So each of the circles correspond to neurons. The connections between those circles correspond to synaptic connections linking those neurons together. So this network in this naive state, if given any particular input, you can equally expect any given output. Each output is equally likely because the connection strengths between those neurons are uniform in nature, or they're randomly set. But when a certain experience happens and learning happens, then that might lead to the strengthening of connections amongst, for example, coactive neurons which were coactive during that experience. It might lead to the weakening of connections amongst other neurons. And the result of that is that now if you give it the same input, or even a partial version of that input, it's more likely by chance to reproduce the original output pattern. So this idea comes probably originally from Donald Hebb's writings in the late 1940s. He was a Canadian psychologist based at McGill, and had ideas about cell assemblies and so on. And about 20 years after he sort of wrote about cell assemblies, then Bliss and Lomo came up with evidence for a mechanism that corresponded to this idea that you could strengthen connections between neurons. And this mechanism became known as long-term potentiation, or LTP. Then about 10 years after that, Masao Ito, Mark Baer, Serena Dudek, and others identified a mechanism which sort of did the reverse, that in an activity or experience-dependent way, led to the weakening of connections between two neurons. And that process became known as long-term depression. And then more recently, in this century, then people have used approaches where you can label active neurons to show that those active neurons that are active when animals are learning something are reactivated when the animals are retrieving, remembering that specific information. Okay, so what about transience? So if stable changes in connectivity promote persistence, they're involved in making the memory, then forgetting occurs when modified synapses are destabilized. And one example of that would be reversing learning-induced synaptic potentiation, synaptic strengthening, would lead to a reduced probability of the former brain state reoccurring and produce forgetting. So that's the sort of conceptual framework for how we might, from a very simple point of view, think about forgetting, is that we're undoing the work that was done in making the memory. So one process that happens during making a memory is, as I mentioned, the strengthening of synapses via like an LTP, long-term potentiation type process. And one of the mechanisms involved in long-term potentiation is trafficking of receptors, AMPA receptors specifically, in the postsynaptic membrane. So this shows the presynapse, the postsynapse, and during learning, these AMPA receptors are moved into the membrane, and that makes the postsynaptic cell more responsive to a presynaptic input. It's going to fire more. So you might imagine then a very simple idea would be that perhaps forgetting involves exactly the reverse of that process. And that is the removal of these AMPA receptors from that postsynaptic membrane. So this is work that's done by Yutian Wang. He's at UBC. And he tested this idea by studying first long-term potentiation. So he did this experiment in rats. And in a long-term potentiation experiment, you stimulate with high-frequency stimulation, and you record from the postsynaptic neurons. And if you do that high-frequency stimulation, that leads to a potentiation, shown here on the sort of y-axis. But you can see in these red and blue labeled groups that potentiation declines over time. It decays back to baseline after about 2 hours. What he did in this experiment, though, was then to give a drug which prevents the internalization, the removal of those AMPA receptors, and ask whether that would block the LTP decay. And you can see in the green lines here and summarized in this graph over here on the right, that indeed did prevent the LTP decay. So that's very suggestive that this might be involved in forgetting, because we think that LTP, the strength of synapses, is involved in making a memory. If we prevent that from decaying, then we might be able to prevent forgetting. And that's what he tested in his rats in the next experiment. So what he did in this experiment was to train them in a task. It's called inhibitory avoidance. They learn that if they enter a certain room, if you like, and a bad event happens, they get a mild foot shock. The trick he did in this experiment was to train them weekly. So they only got a very, very mild foot shock, and they can show memory for that foot shock in this sort of red bar when you test them 1 hour afterwards. But if you test them 24 hours afterwards, then their memory seems to have disappeared. So it's like a short-term memory. Then he asked, what happens if he then injects them with drug that prevents that removal of the AMPA receptors, that prevents the LTP decay? Can he now make that memory last and prevent the forgetting? And that's indeed what he sees. So that's that group over here. So that suggests of one mechanism that might underlie forgetting. So you can weaken synaptic connections, but you can also remove synaptic connections, and presumably that would do exactly the same thing. If you remove a synaptic connection that's involved in a key memory, then presumably that makes that memory harder to access or weaker, degrades it in some way. And this idea was explored by Kasai in this paper. And he did this in part of the brain involved in motor memories. So the types of memories that we have for like how we walk or how we ride a bike or how we ski and so on. And so he trained his mice in not in a skiing or cycling, but just on a sort of treadmill type apparatus. And so they formed this motor memory. They could learn efficiently how to walk on this treadmill. And then the trick he did here was then he developed a very clever genetic approach which allowed him to specifically eliminate recently potentiated synaptic connections or spines. And so then he applied this drug to wipe out the recently potentiated spines, which presumably correspond to the motor memory that the animals had just formed. And when he did that, then he saw complete forgetting of that memory. So the animals went down to baseline, to the state they were at before they had learned anything. And so in addition to weakening synaptic connections or eliminating synaptic connections, you can also imagine that doing the opposite, adding synaptic connections, might also lead to some forgetting, because you're rewiring the brain that might make a memory that's stored in those circuits a little bit more difficult to access because you've messed up with the wiring diagram, essentially. And that's the process I'm going to talk about in a bit more detail now. So that process is hippocampal neurogenesis. So why hippocampus? Hippocampus is a part of the brain which is involved in the types of memories that we sort of most naturally think about, the memories for events like our birthdays, like when we got married, those things and so on. And neurogenesis, well, neurogenesis is just simply refers to the process of generating new neurons, neurogenesis. And that happens, of course, during development. That's how our brains are made. But it persists in just two parts of the brain during adulthood. And one part of the brain is the hippocampus. The other part I'm going to come to later is the olfactory system. So in the hippocampus, what I've shown here is a cartoon of one area of the hippocampus where neurogenesis happens. This is the dentate gyrus of the hippocampus, and these neurons are sending projections onto neurons in another region of the hippocampus, CA3. So these neurons gradually integrate. So you have neural progenitor cells, they divide, give rise to new neurons. These new neurons send dendrites up towards it's called the molecular layer, that allows them to receive inputs from the cortex. At the same time, they send an axon towards the CA3 region, sort of hooking them up with other parts of the hippocampus. And then over the course of weeks, you get continued growth of that dendritic arbor, meaning they can integrate lots and lots of signals from the cortex. You get synapses form onto output structures in the CA3 region. And during this period, these new synaptic connections are coexist with or even replacing existing synaptic connections. So you've got this very dynamic situation in the otherwise relatively stable brain where you're adding new neurons, they're receiving inputs, they're making new output connections, and at the same time, they might be eliminating existing connections. So given that this sort of dynamic situation, you think, well, that might mess up with stuff that's already stored in the memory, and that's the sort of idea that we had. So this is Catherine Akers, who was a postdoc in my lab, who, along with Anne Wheeler and Alonso Martinez-Canabal, did these experiments. And so what they would do would be to take their mice, train them in a task which they believed would engage this hippocampus structure, and then they would introduce some manipulations that elevated hippocampal neurogenesis. And I'm just going to talk, I think I'm going to talk about all three of them, but I'm going to start by talking about a very simple manipulation, an environmental manipulation that robustly increases hippocampal neurogenesis. And that manipulation is exercise. And so if you take rats or mice when they're housed in their cages in a university laboratory, and if you stick a running wheel in their cage, then they will spend lots of hours per day running on that wheel. And you see this, of course, with pet hamsters and so on. And the consequence of that running is that changes the brain in a number of different ways, one of which is it increases neurogenesis, the production of new neurons in the hippocampus. And so you can see here what we did in this experiment was to give mice access to a running wheel. They run about 5 or 6 kilometers per day each. And then we took their brains out at different time points, and we measured markers that were suggesting that cells are dividing. So you need cell division to make new neurons, and also markers of new neurons. This is a double courting. And you can see within just a few days of initiating the exercise, you get this ramping up of neurogenesis and increase in the production of new neurons, whichever way you measure it. So then Catherine could ask, what happens if you do that after training? So the prediction is if you increase neurogenesis after training, you're rewiring the circuits where the memories are stored, and maybe that leads to forgetting of those memories. So she did this experiment shown here. So she took animals, mice, and trained them in contextual fear conditioning tasks. So this is another task I'll just briefly explain. What happens is the mice get placed in a place they've never been before, an operant box. After some time, something bad happens. They get a mild foot shock, And then if you place them back into the same context, same place, a day later or a month later and so on, then if they recognize it, they're going to act fearfully. They're going to say, oh, this is the place where I was shocked. And one way in which they act fearfully is that they freeze. So basically they stop moving, they adopt this crouched, motionless posture. And we use that as an index of how strong their memory is. So what we wanted to do in this experiment then was to have the learning event, train them here. Then one group is housed normally, the other group gets a running wheel for 6 weeks, and then we test their memory here. And the prediction is that this should increase neurogenesis and produce forgetting, and that's indeed what we see. So this reflects in the running mice that we see an increase in the production of new neurons in the hippocampus, and at the same time, it looks like we've weakened that contextual fear memory. So that's great news. It's consistent with our hypothesis, but there's many other possible explanations. You might say, well, the animals that exercise, they're just less anxious, and the less anxious animals are just not going to do this freezing behavior. And so one way in which we control for that is simply reversing the order of learning and exercise. So in this group, instead of learning first and then exercising, they now exercise first and then learn, and then we test them here. And the important thing is that the temporal relationship between our manipulation, the exercise, and the test is the same in the two experiments, just the order of learning and exercise is reversed. So as expected, you see the same increase in neurogenesis, the same increase in the production of new neurons. But at the same time, you don't see any forgetting when that increase precedes the learning event. So it's only in the what psychologists call the retrograde direction are we seeing these forgetting effects. So we went on to then just ask, well, people would say, well, running is a very non-specific way of increasing neurogenesis because that does many things to the brain. Maybe one of the other things it does to the brain is producing your forgetting effect. And so we used sort of increasingly more precise ways of manipulating neurogenesis. This is a genetic method. And you see that you get the same result. That is, you can increase neurogenesis and it produces forgetting of that contextual fear memory. This is a pharmacological method. Again, we can increase neurogenesis and we decrease the strength of the memory. So this is a graduate student in my lab. He was interested in, well, what's driving that effect? Why is this happening? And of course, our hunch, and I've sort of mentioned it already, is that we think the rewiring that happens in the brain as a consequence of neurogenesis is essentially incompatible with stable information storage, stable memory storage. And so I wanted to show you this just because it's a nice picture of the dentate gyrus showing the granule cells and their dendrites where they receive inputs from the entorhinal cortex, from the cortical inputs, and then they send this axon towards, known as a mossy fiber, towards the CA3 region where they form these large synaptic connections called large mossy fiber terminals. And so what Axel reasoned was forgetting might be occurring in our studies because we've just got many, many more neurons, or because we're changing connectivity, or some combination of those two. And what he wanted to do then was to discriminate between those two possibilities. And what you'd want to do, of course, is to change one of those, for example, the way in which new neurons connect into the network without changing the other factor, that is the numbers of new neurons. So I'm going to just show one experiment here, but what he did in these experiments was to take advantage of genetic tricks which allowed us to manipulate how the cells are integrating into the circuit without changing the numbers of those new neurons. And I'm going to talk about just this hyper-integration or sufficiency experiment here. So what he reasoned was that, okay, we don't change numbers of new neurons, but we make them become really, really integrated. So they get many, many more inputs, they get many, many more outputs onto CA3 pyramidal cells. Maybe that alone would be sufficient to produce forgetting without changing overall levels of neurogenesis. And so in this case, he used a recent sort of trick which has really revolutionized neuroscience, and that is the ability to manipulate the activity of neurons and cells with light. And so what what he's done in this experiment is to express a light-sensitive channel rhodopsin, only in newborn neurons, the ones that are shown in green. And then when he shines a laser on those neurons, he can activate those neurons. And the particular manipulation, people normally use this approach to fire mature neurons, but he wanted to see whether he could use this approach to make them grow faster, essentially. And that's what he did in this experiment here. So you can see that compared to the control neurons shown here, the ones that received the light stimulation seem to have more complex dendritic arbors. They've grown faster, they've integrated more. They have more dendritic spines, and so these are the points of contact where synapses are forming. And then they seem to have more connections, more large mossy fiber terminals in the output region as well. So basically, evidence together that just says these neurons are becoming hyper integrated in this channel rhodopsin group. So then you can ask whether that's sufficient now to produce forgetting. And indeed it is. So what he did is to combine the experiment I just described with the behavior experiment. So he trains them, they learn about this context. He then stimulates those cells 15 days, once a day for about 3 minutes, and then test their memory either 15 days later, right after the stimulation ends, or 30 days after the training in this case. And what I should mention briefly is that when these new neurons integrate into the brain, they don't form synapses right away. They take about 15 days before they form synapses. So he reasoned that if it's the process of synaptic integration that's producing forgetting, then you shouldn't see forgetting in this group here, where you test them right at the 15-day time point, because those new neurons haven't had sufficient time to essentially rewire the circuitry. And that's indeed what he sees, no forgetting in that group. But it takes the same animals and waits another 15 days, then you see the forgettings happen. And that presumably is because you've given the brain enough time for those manipulated neurons to then become integrated and sort of mess up the circuitry in the hippocampus and produce forgetting. So that's a summary of I just showed you one of those experiments, but we've done a bunch of different experiments here showing that the same class of manipulations that produce this hyper integration phenotype is sufficient to produce forgetting, and then restricting the amount to which these new neurons integrate into the circuit can block the exercise-induced forgetting effect. So one question we then became interested in, and I mentioned this earlier, was that neurogenesis in the adult brain in rodents happens in two places. One is the hippocampus I just mentioned, and that's important for forming and recalling these memories for events. And then the other place is the olfactory system. And this shows here the olfactory bulb. And so Jana Hasse in my lab, who was a postdoc, was interested whether a similar sort of turnover process of new neurons in the olfactory system might lead to forgetting of olfactory memories. And the way in which she did this was to use a olfactory reward-based task. So in this task, I'm going to show you a video of it in a second, but just to describe the sort of principle is the animals get to dig in these bowls of bedding, which are scented with particular odor. We call them odor A and odor B here. When they dig in the odor A bedding, it's reinforced. They get a sugar pellet. When they dig in the odor B bedding, they get nothing. And so over time, they should develop a preference for odor A over odor B, especially if they're hungry. And so this is what it looks like in practice. You can give them a probe test where you ask them, which one do you prefer, odor A or odor B? And you can see in this case, this animal that's been trained spends most of its time digging in the previously reinforced odor. So it's remembered that that was the odor that was associated with the sugar pellet. So that's the type of experiment that Jana did. She trained them in this A versus sort of B discrimination, very simple discrimination. She increased neurogenesis using similar methods to the ones I described earlier. And then she tested their preference in this probe test. And what she found was that with the pharmacological method of increasing neurogenesis as well as genetic method. Basically, you get the same effect. You can produce forgetting of these odor memories. Okay, so this is a summary of 3 ways in which we think the brain can forget. So you can weaken synaptic connections from previously potentiated connections, and that produces forgetting. You can eliminate connections, and you can add new connectivity, and that also interferes with the ability to retrieve that old information. So this brings up the question, why should we forget? Is it important for anything? And so I think colloquially, when we talk about what we think about forgetting, we think of forgetting as a bad thing, as a sort of failure of our memory. But it's long been sort of embraced by psychologists, especially, that certain amounts of forgetting are probably useful for our brains. And so this is William James. He was a psychologist at the turn of the last century. He talks about it as being in the practical use of our intellect, forgetting is as important as remembering. And then around the same time, and very much in the same vein, the French neuropsychologist Ribot came to this conclusion. He said that we thus reach the paradoxical result that one condition of remembering is that we should forget. Without totally forgetting a prodigious number of states of consciousness and momentarily forgetting a large number, we could not remember at all. So oblivion, or forgetting, except in certain cases, is thus no malady of memory, but a condition of its health and its life. So inspired by Ribot and by James, what I want to spend the second part of the talk on is this idea that forgetting is adaptive, and that is adaptive in the following 2 ways: that it optimizes memory-guided decision-making by first, in changing environments, forgetting is useful since it removes outdated information and allows more flexible behavior. And in noisy environments, forgetting is useful since it prevents overfitting to specific past events and promotes generalization. So I'll get to that in a second. So first, in changing environments. So some of you may recognize this picture. This is Yorkdale Mall. And when you go to Yorkdale Mall, you might park your car in the parking lot here and then go visit the mall, come back after a couple of hours, and then you've got to remember where you parked your car. And then the next time you visit, you might park in a slightly different spot. And then the third time you visit, you might park over here. So the trick is, when you're going back to look for your car, you don't want to be remembering on the third occasion where your car was on the first or the second occasion, because that's one, it's irrelevant, and it's going to interfere with your ability to retrieve this memory, the correct place, location of your car. So we wanted to test that experimentally and in the context of manipulating neurogenesis. And so we came up with these predictions, and that is that insofar as increasing neurogenesis, as I've just described, should produce forgetting, should reduce the probability of successful retrieval of a learned memory. At the same time, as you've weakened that initial memory, maybe that facilitates encoding, learning of new information that conflicts with that original information. And so in psychological terms, that would be decreasing proactive interference. And then the other opposite version of the experiment would be as follows. So what if you decrease neurogenesis, so reduce the amount of rewiring of these hippocampal circuits? That should stabilize memories stored in those circuits, should increase the probability of retrieving those memories successfully. And insofar as you've stabilized, or if you like, made stronger those original memories, maybe that would interfere with encoding of new information, especially information that's conflicting with that new information. So it's hard to learn 2 things which don't fit together. And that would be the case there of increasing proactive interference. So this is Jonathan Epp in my lab, who was a postdoc, and he used the water maze for these studies. If I can, I was hoping to show you the video here, but it's not going to play. But in the water maze, let's see if I can get a bit of this. Yeah, so I can play it now. It's great. Okay, so the water maze is a way in which we can test spatial memory in mice. And so what happens? You place the mouse in that pool. The water is made opaque with white paint. It can't see that platform, which we can see from above. And its job is to try and find that platform so it doesn't have to finish swimming. So at the beginning of training, you can see it sort of swims around randomly. It's not very good at finding that platform location. At the end of training, boom, it gets there right away. And then you can ask the mouse, where do you think that platform is, in something called a probe test, where you trick the animal and you remove the platform from the pool, and you measure where the animal searches for it. And you can see, in this case, this well-trained animal has a very good idea about where that platform location is. Okay, so what Jonathan did then was train the animals in that type of task. That they learned the platform was here in the northwest quadrant of the pool. Half the animals exercise for a month, the other half are sedentary. This one obviously increases neurogenesis. So we test that original memory. And then we move to the second phase of the experiment, where we ask them to learn something new. We move the platform to the opposite part of the pool. So obviously in conflict with that original memory. So this simply shows that we can increase neurogenesis with our manipulation. And when it happens after training, we can reduce the strength of memory. So just like the experiments I showed before. So this is the amount of time the animal spends searching where the platform is. And you can see it's reduced in the runners compared to the sedentary mice. So they've gotten to a certain degree that spatial information. But when you now train them in this reversal task, the running mice are much more readily able to learn the new platform location. So having a weaker original memory frees them up to learn the conflicting information in the second phase of the experiment. You can do the opposite, and that is to reduce neurogenesis and ask whether it does the opposite, and that's shown here. So in this experiment, in our control mice, you can see this natural forgetting. So comparing comparing this bar to this bar, you see there's some forgetting over that 42-day delay between the end of training and testing here. But if you block neurogenesis, you can see relative stabilization of that original memory. There's less forgetting in that group. And the animals with blocked neurogenesis, they have a stronger original memory, now have more difficulty finding the platform in its new location. They have more trouble with this learning this conflicting information. The original memory proactively interferes with encoding of this new information. So Jonathan wanted then to see if this generalized to another type of learning. So in this case, he did it with this task here. So they are trained in, it's another olfactory-based task, but it's a lot more complicated and importantly depends upon the hippocampus. So what they've learned, two things concurrently. When they're in this context, the white context, they dig in the bedding that's scented with cinnamon, they get reinforcement. If they dig in the coffee bedding, they get nothing. At the same time, they're trained in the stripy context and they learn the opposite. The coffee is now reinforced, cinnamon is not. And this is, what it looks like in training. So this is the mouse digging and then in a second getting reinforced with a reward. Okay, so that's called paired associate learning. So they're making an association between a context and an odor. And if we think about those as algebraically, you can sort of think of them in shorthand as A1, B2. That's what they're learning in the learning phase. Then we can increase neurogenesis. We can test them on those original paired associates, A1, B2, see if we see any forgetting. And then we can train them in the reversal of that. That is, now they have to learn that coffee, which used to be reinforced in the stripy context, is now reinforced in the white context. So it's quite difficult. They've got to reverse what they learned before. So sort of focus on the two left-hand bars in each of these graphs. So this is the end of training. They learn the task very well, almost 100% correct. Increasing neurogenesis after training makes them forget in the green group compared to the blue group. And then in the green group, the ones that have forgotten now can learn this A2B1, this reversal phase of the experiment, much better than all the other groups in this experiment. So it suggests that, yes, forgetting the old information, which has become outdated in this case, allows you then to more readily, more quickly encode the new information. So it's really good if the environments are changing, which of course environments change all the time. Contingencies are always changing in our world. So that's the sort of conclusion. But you might say, well, maybe we've increased neurogenesis in these experiments. Maybe it's just that they just learn better. So it has nothing to do with the degree of conflict. So what you want to do is do an experiment where you manipulate the degree of conflict between in these experiments. So again, exactly the same as before. They learn A1, B2. We increase neurogenesis or not. We test them on those original paired associates. And then we have a high interference group. They have to learn the reverse, A2, B1, or a low interference group where they learn completely new paired associates, completely new contexts, completely new odors. And you can see that only in the high interference group is there an advantage for the runner. You don't see any advantage for the runner in the low interference group. What that suggests is that it is dependent on the conflict between the new information and the old information, that wiping out the old information helps the new information to be encoded that's conflictual with it. So similar findings have been found in flies. So very quickly, because I think I'm running out of time here a little bit. This is a study from Zong's lab, and they did a very similar task. So in this, they paired odors with shock. So odor A was paired with a shock, odor B was not paired with shock in this kind of experiment. And the flies have to learn then to avoid odor A and spend more time around odor B. And what they discovered in the process of their studies was they found a fly that didn't forget, or at least forgot more slowly, this one. This is a fly which expresses, it doesn't really matter, but a dominant negative RAC1. And then they went on to say, okay, what happens now if we do the same type of experiment that I just described, where you reverse that? So they first learn that A is paired with shock and B isn't, and then the next day we pair the B odor with shock and the A isn't, and then you reverse it again, and so on and so forth. And the animals that, or the flies in this case, which have slower forgetting, have an inability to reverse. Basically, they're remembering the first memory too strongly, and so they can't overwrite that memory in the second when the contingencies are reversed, and that's shown in these graphs here. They also, in flies, you can do many great tricks. You can also make flies that also forget more rapidly, and that's what they did in this experiment, and they showed that the sort of reverse is true. The more the flies that could forget more rapidly could sort of keep up with the reversal learning much more efficiently than the normal flies. Okay, so the last part of my talk is about the second sort of benefit of forgetting, and that is that it might prevent overfitting and help generalization. So here I've got a quote from computer scientist Jeff Hinton, of course, from U of T. And what Jeff says is the brain has about 10 to the 14 synapses, and we only live for about 10 to the 9 seconds. So we have a lot more parameters than data. So the point he's making here is that the issue in the brain is perhaps not a lack of space. We have plenty of space to store as memories as we want. The real issue is using up too much space to store any individual memory. That is the process of sort of essentially overfitting information. So overfitting is a big issue in machine learning, of course. And so it's illustrated here. So what we have, we're trying to fit a model to the data points shown here in blue, and we're going to use that model then to predict the new data points shown in green. So if you use a regression or a model which has high number of parameters, you can fit those blue data almost perfectly. But that model is not going to be very good necessarily at predicting the new data points shown in green. So it's overfitted essentially. You could alternatively use a much simpler model, a regression with fewer parameters, that fits the blue data points pretty well, but is way better at predicting the new data points, the unseen data shown in green. So you can think about this if you, as John mentioned, I'm a big soccer fan, so I've probably kicked many, many soccer balls in my life, all of them very, very different. And it makes no sense that I would remember every single soccer ball that I have kicked. But what it does make sense is that I would abstract from those soccer balls some sort of general principles, like that soccer balls are typically made out of interlocking hexagonal or pentagonal sort of pieces of leather, for example. So in machine learning, there's a number of techniques which people use to prevent this problem of overfitting and promote simplification of models. And so some of those are listed here: dropout, sparse coding, weight decay, weight elimination, noise injection. And what struck Blake Richards and I was that at least some of those, if not all of them, correspond to things which we've seen in the brain, which are mechanisms of, or potential mechanisms of forgetting. So weight decay, the weakening of connections between neurons or units in your model, your neural network, seems to correspond pretty nicely with that depotentiation AMPA endocytosis or AMPA removal process I described at the start. So the way of weakening synaptic connections. Weight elimination, what might correspond to the spine shrinkage experiment that I described, where if you remove the recently potentiated spines, you induced complete forgetting. And noise injection, you could easily imagine that that would correspond to the neurogenesis process. You're adding these new noisy hyperexcitable units into a circuit, the circuit gets rewired, that's some form of noise injection. So this is Blake Rich, as I mentioned him already, and he wrote the review with me, which is forming the basis of this talk. And he was interested in the relationship between forgetting and preventing overfitting and generalization. And so we go back to Yorkdale Mall. You've now been there many, many times. And at this point, it's certainly not adaptive or useful to remember every single parking spot you ever parked in the mall. Indeed, we none of us really do remember that information. What's much more useful is just to remember perhaps that parking here in the northwest part of the parking lot is normally the place where you find the best parking spots. So you formed some kind of general model, you've forgotten the details of each parking spot that you've gone through. So that's the idea. So he's interested in to what extent does forgetting detailed information allow for the emergence of a much more generalized model, and that generalized model can then be used more usefully to predict the future, to predict future data points. So he did it using a water maze, like I described before, but it was a slightly modified water maze. So instead of training the animals on a single platform location, he trained them on each day on a different platform location. And those locations were drawn from a Gaussian distribution. So on any single day, they have no information about the overall distribution, but over the course of training, the overall distribution, they can integrate that information and become knowledgeable about that. So he was interested in two things. To what extent do the mice trained this way forget the detailed information, that is the individual location of all these different platforms? And then secondly, and to what extent do their searches match the overall distribution from where those platform locations were drawn? And he hypothesized that not only should the animals acquire that through the course of training, but also in the post-training consolidation period, then that might enhance this process of abstraction or generalization. So this is what the platform locations look like in his experiment. That's the distribution from which they were drawn. That's the training data. They can learn this task. It takes about 9 days to train them on each platform location each day. And then he tested their memory in that probe test, like I showed you before, 1 day afterwards or 1 month afterwards. And he was interested, as I mentioned, in two things. To what extent do they search the exact platform locations from before, that the detailed memory, have they forgotten the details? And at the same time, are they better at matching the distribution from which the platforms were drawn at the more remote time point? So he found, yes, indeed, there was some forgetting of the details. So the mean platform crossings is shown here at the 1 day time point, 30-day time point, 30 days afterwards, yeah, they've forgotten. Their search seems a little bit less precise, at least in terms of the exact platform locations. And then indeed at the 30-day time point, there's this better match between their swim path distribution and the distribution from which the platforms had been taken. And so what essentially this is telling us is that, yes, they've lost information about the trees, but they're now seeing the forest. So they have a much better, more general model, which is probably more useful in the future. So I see that it's 2:57. So I'm just going to skip this very last part of my talk and just come to the conclusion. And it's a shame because I asked my graduate student to prepare these slides at this last part of the talk this morning. He did it in about half an hour for me just before. Anyway, so what I just want to wrap up here and say, we've been interested in the neural mechanisms of forgetting. I've talked about here some of those, synaptic weakening or elimination and circuit remodeling. There's others as well, which I'm happy to talk about. Forgetting, at least partial forgetting or low levels of forgetting, is not necessarily a memory flaw. It may make you smarter. And it may make you smarter in two ways: that improves your flexibility in changing environments by getting rid of outdated information. Our world is always changing, becomes decorrelated as a function of time, so that should be useful. And secondly, forgetting allows more efficient generalization, as I just described, by preventing overfitting to specific past events. And with that, this is, we call this the selfie slide. So once a year in our lab, we ask everybody to take a selfie of themselves, and this is the last version of that event. So all the people in the lab have contributed to this work. So with that, I'll stop there, and I'm happy to take questions.